Good morning, and thank you for inviting me for that talk. So I will talk about Cardio3 BioSciences, which is a publicly traded company in Brussels and Paris. We are today active in two therapeutic areas in cardiovascular diseases, where we are pretty advanced. C-Cure is our program there. It's an autologous cell-based therapy that is, we reprogram cells to become cardioreparative and then inject them into the heart of patients. And then we have a CAR-T program, which we've acquired, and I will also talk about that as well. Obviously, all those are modified cells, so it is covered by strong IP. We have our own GMP facility, which I think is also an advantage in this space, looking at the complexity of cell manufacturing, of course. And we have multiple short-term catalysts. So this is our pipeline. The first two programs, the Chart-1 program is a European phase 3 trial currently ongoing. We have completed enrollment in that trial, and we will have a futility analysis in the next few days. And then the primary endpoint of that trial should be read in around mid of next year. Chart-2 is the US version of that phase 3 trial, which has been cleared by the FDA, although it is currently being reassessed as we are changing our delivery system, and we will wait to hear until the FDA clears us to use that delivery system to use in this trial. In the CAR-T space, we are in phase 1/2A on AML and MM. We are currently enrolling patients, and we're looking to progress those programs both on blood tumors as well as solid tumors over the course of next year. We have an allogeneic platform as well, and two other receptors. Again, I'm not going to spend too much time on that, but I think that those are of interest. So this is our Chart program, which is the phase 3, as I said. We're enrolling neocardioception class 2B, 3, and 4A. The follow-up is at 9 months. It is a composite endpoint looking at mortality, worsening heart failure, 6-minute walk test, and quality of life, and then cardiac function. As I said, it's currently running in about 12 countries. And Chart-2, I've talked about that. The current clinical endpoint is 6-minute walk test. This is what the FDA has approved as a primary endpoint. However, we are discussing with the FDA to try to include some mortality and morbidity elements into that endpoint as well. FDA seems to be more open now to composite endpoint than they were at the time we first discussed with them that trial. In our CAR-T program, we are using NKG2D, which is a receptor of NK cells that are transduced onto T cells. It has very particular methods. It's not another antibody. It is actually a naturally occurring receptor on the NK cells. It is an activating receptor of those cells, and the killing activity is triggered by changes in the cells. Many tumors express NKG2D ligands. You can see here about 80% of all tumors are expressing NKG2D ligands, which again is pretty unique. This is, to our knowledge, this is one of the only CAR-T technology that allows to target many, many different types of cancers. So that's very exciting for us looking in the future. The preclinical data on this was also interesting and intriguing. We've done that on 5 different tumors in mice, and you can see in all the cases 100% mortality versus 100% survival for treated animal. But even more interesting, once you have re-challenged the surviving animal with the same tumor, you actually get all the animals that survive again, which is due to the T-cell memory effect because the CARs at that time have disappeared. So this is, again, a pretty unique mode of action that targets, as you can imagine, very well the metastasis world. In terms of news flow, we have a pretty dense news flow over the next 18 months, which both on the cardiology program as well as in the CAR-T program. And in terms of corporate highlights, quickly, we've had $41 million of cash as of September 2014, and our stock did pretty well. We have multiple near-term catalysts. We're obviously very focused on US now because of being that being the main market, both for capital as well as for healthcare. Thank you.
Great. Thank you, Christian. Please come join me, and I think there's a speaker or a microphone right behind your chair. So that was a good overview, and as you mentioned, your lead program is known as C-Cure, which are autologous cardiopoietic cells, specifically to treat ischemic heart failure. How are these cells delivered? And maybe you can tell us sort of, you had mentioned that there's some changes going on with respect to the protocol for chart 2. Maybe you can go into that a little bit.
Sure. So we are using our own catheter for delivering those cells, which is called C-Cathez. And we designed that catheter because we've seen with many other trials, including ours and other people trials, that the number of cells that remain in the heart after the injection is pretty limited. And if we want to have a more efficacious product, we needed to have that retention rate, or goal retention rate, increase significantly. So we designed a catheter to do just that. And this is the catheter that is used in our phase 3 trial in Europe. In the US, as at the time we filed the IND, we had no human experience with that catheter yet, so the FDA was reluctant to grant us a phase 3 status with that catheter. Now that we have human data, we hope that we'll be able to get there.
Great. And then maybe you can walk us through some of the phase 2 clinical benefit in terms of what you've shown with C-Cure. What is the effective dose that you're taking forward in the phase 3 studies? And what benefits did you see in terms of cardiac function, in terms of structure, and also exercise capacity?
Sure. So the dose we're using is 600 million cells. Again, it's very difficult to determine a precise dose in clinical trials in cell therapy. Our floor is 600 million cells because the retention between patients and patients, between injection and injection, varies so much that having a real number of cells that is active is a very difficult exercise. However, we've seen in our phase 2 trial, we had between 600 million cells and 1.2 billion cells injected, and we've seen that with 600 million cells, we had efficacy. So we moved on with that dose. The second thing on the outcome, yes, we had ejection fraction improvement of about 25% relative, so 7 points of ejection fraction on the treated patients versus the non-treated patients, which was good. We also saw reduction in the volumes, systolic volume and diastolic volume, which was systolic was also highly statistically significant. Exercise capacity as well. We had 77 meters difference between treatment and control. Mortality, although you do not talk about mortality on such small trials, but also everything was going in the same direction. When you do look at those small trials, it's very important that you do focus on seeing a coherence between the data, between all the different parameters. If you have ejection fraction increasing by 25%, and at the same time you have walk distance decreasing by 10%, that's not coherent. So you need to see an improvement that is coherent among all the endpoints.
Great, that's helpful. Now these are autologous cells, so cells from the patient. So how do you process the cells? Maybe you can walk us kind of through the process in terms of when they first come in and how you handle the cells. And are you doing any forms of manipulation to optimize these cells?
Yes, so we take the cells from the bone marrow of patients. We send a specific kit to the hospital in which that bone marrow is put in. It is sent to us at a low temperature, but not frozen. We get them, we do QC, and then we process the cells for about 30 days. 23 to 27 days to be accurate. In that process, we expose them to a number of factors that are the same factors in embryological development that makes a cell, an embryonic cell, become a heart cell. And we use those same factors, and the cells are different. They are expressing some markers of cardiac differentiation, and they have been shown to be very effective in animal models before we took them into humans. So indeed, I mean, and then we freeze the cells at the end of the process at minus 160, and then we send them to the hospital when the patient can receive his therapy. So that knowledge that we acquired moving from phase 2 to phase 3 in the development of autologous cell therapy is of incredible value to us moving into our CAR-T program, because that's very similar in terms of approach.
Yep, excellent. I can certainly see that, and I definitely want to pick up on the CAR-T program. But since you have the phase 3 studies ongoing right now, and you gave a good overview about when to expect enrollment and when to expect data, enrollment to complete and data, what is the regulatory strategy, kind of between the U.S. and Europe, because the endpoints are a little bit different? Is there a risk that maybe one trial works while the other doesn't? Do you need to show both studies? Just at a kind of higher level, walk us through sort of some of the planning that went into designing the phase 3 studies and the thinking to try to cross between the two regulatory bodies, if you would.
It's a very difficult to answer question, because ideally we would have done one big trial, US and Europe. However, the agencies in Europe and the US did not agree on what the endpoints of those trials should be. So instead of fighting, we decided just to do two different trials. And one of the reasons why we'd like to use our catheter in the US trial is also to be able to pool the data from the two trials, because apart from that, they're identical trials.
Exactly. And what, again, assuming positive data readout, again, I think this will actually be one of the more important readouts for the cell therapy sector in the next 12 to 18 months, and with a futility analysis coming sooner than that. How does Cardio3 anticipate commercializing C-Cure?
Commercialization is always going to be, for cell therapy, we believe, into centers of excellence. It's never going to be very broadly distributed, because it does require specific skills for injection as well as preparation of the cells and things like that. So we believe this is typically the type of therapies that we, as relatively small company, can certainly undertake ourselves. However, the probability that pharma company would come at the time of positive phase 3 data is significant. So we will see at that time if we will continue pursuing that commercialization strategy ourselves or not.
And it was interesting in the panel, that we had this morning, we talked about how different it is to commercialize cell therapies versus sort of how big pharma is oriented today. Excuse me. Do you think that there are different types of companies or different groups within pharma, or do you think pharma needs to make some changes to deliver these advanced therapies, or do you think that this will, in particular with C-Cure being kind of a add-on into the heart failure space, be maybe a bridge to move into how we commercialize advanced therapies?
Well, it's basically a hybrid model. I come personally from, my career was spent on the device side, and on the device side you have less sales reps and more what was called field clinical engineers. So people that were helping the physicians to actually do the therapy, whether it was implantable defibrillators, biventricular pacemakers, and so on and so forth. So we believe that cell therapy is going to be something similar to that, in which it's going to be more education, guiding, than it is going to be a pure sales function. From a logistics perspective, it is more challenging than a typical pharmaceutical product, but that doesn't mean that it cannot be done. Many people have started doing it in the past. We've learned from some of the mistakes that were done. I'm sure we're going to do our own, for sure, but at least we're not going to repeat the same mistakes that others have done.
That's actually a really helpful perspective. So switching gears to the CAR-T approaches, you mentioned that you in-licensed some of this technology from Harvard. Tell us a little bit more about how these natural killer receptor CAR-Ts differ from some of the other technologies out there. And what I find really interesting about your approach is it really is a pipeline of CAR-T products. So maybe you can kind of comment on how these individual therapies are even different within your own pipeline.
Sure. So what we use is a receptor that is present on NK cells. The nice thing about it, it is a naturally occurring receptor. So it doesn't have to be adapted or spatially, geometrically adapted when you transduce it onto a T cell, and it binds naturally to the natural ligands of NKG2D. So it's quite easy in terms of binding affinity and all of that for treating tumors. The very nice thing about it is that with that one particular receptor, you can target many different cancers as well. So once you've passed your safety data with your phase 1, then you can move into many, many different phase 2 data without having to repeat. So that's very different from using antibodies like CD19 or others, where every time you go for a new target, you're going to have to repeat your phase 1 to make sure that your safety is good, and then move on to the next product and next product and next product. This is a unique approach to CAR-T. Obviously, it's early, so we need to see how we pass the phase 1 outcome, but it is very exciting.
And we have about a minute and a half left, but when should we get data from the first hematologic phase 1? I think it's in acute myelogenous leukemia and multiple myeloma. When could we get data from those first cohorts?
Well, those are open label studies, so we are going to be getting data basically every quarter. Every time we finish a cohort, then we get to the result of that cohort and then go to the next cohort. We're currently running the trial in one site. We put now one at the Dana-Farber. We hope to maybe open a couple of more sites to speed up enrollment. But certainly we're going to hear something every quarter from us.
Okay, great. And then last quick question, if I may. With respect to T cells, obviously you guys are a cell therapy company, so there is relationships between these. And as you mentioned, iterative learning from cardiac into the T cells. Would you intend to partner the CAR-Ts because its oncology is a little bit different? Or do you think that this would be something that Cardio3 would ultimately like to develop and ultimately commercialize on its own as well?
This is something that typically we'd like to commercialize ourselves. I mean, we've built an infrastructure for manufacturing those cells very efficiently. I think we're the only CAR-T company that has experience manufacturing cells and have experience taking cells from bench to phase 3. So we're very excited with this technology, and we certainly hope to be able to take it to commercial stage.
Excellent. And then you mentioned your IPO last year. Just remind us the ticker and what your current listing is, and do you have plans to access the markets in the U.S.?
So the ticker is CARD for CARD. We've actually just completed the PIPE 2 weeks ago, mostly with U.S. investors. That was vastly oversubscribed. We raised 32 million euros. That was limited by our market cap at the time, 10% of market cap, but it was vastly oversubscribed, mostly by U.S. investors. So it is obvious that U.S. is going to be a place where we're going to be focusing on.
Excellent. Great. Well, I want to thank you very much, Christian, for taking time with us this morning. Very interesting story, and I think emerging as really a leader in the cell therapy space. So I wish you luck with this, and look forward to hearing more from the company as we move forward.
Thank you very much.